The overall goal of this procedure is to comprehensively assess germline chemical toxicity using the nematode Caenorhabditis elegans. This is accomplished by first synchronizing worms by adding a diluted bleach solution to a mixed-stage worm population of the C. elegans PXOL1 GFP strain. This step generates a large number of embryos protected from the bleach by their eggshell. The second step is to generate a synchronized L1 larvae population by letting the embryos hatch in liquid without food. Next, the worms grow to their fourth larval stage, or L4, at which point they are exposed to the chemicals of interest in liquid for 65 hours. The final step is the visualization through fluorescence microscopy of expression of the GFP reporters in embryos inside the uterus of the worms. Ultimately, the proportion of GFP-positive embryos compared to the DMSO control reflects the aneuploidicity of the compounds tested.
The main advantage of this technique over existing methods, such as rodent models or cell culture assays, is that we can assess the aneuploidicity and germline toxicity of chemicals in a whole animal model, while at the same time performing a high-throughput assay. Performing the procedure today will be the postdoctoral fellow, Dr. Daniela Parodi.
The gravid worms for this procedure were previously prepared as described in the accompanying protocol text. Collect the gravid worms from the nematode growth medium, or NGM plates, by washing them with 1 to 2 milliliters of M.